I like it. So we'll do a quick review while those of you that are still coming in start working on the Socrative activity. Just one question this time. Let's talk replication bubbles. And maybe this will help. So I'm going to let me know if you've got questions about maybe 2 minutes about where this goes. So if you imagine a single double helix, both strands base paired, hydrogen bonded with each other. Let's say there's an origin of replication, and we're going to talk a lot more either later in class today or next time about how scientists identify where origins of replication are on chromosomes. This has sort of been a mystery so far in class. I've just been making up saying, okay, here is going to be an origin of replication. A lot of cartooning. We're going to get into the details. Let's say the origin of replication is there. So what's the first thing that happens? The two strands separate at the origin of replication. So the next thing that you would see, if you were watching this happen through time, is that you'd see a small bit of the double helix that's unwound. So the hydrogen bond donors and acceptors of the nucleotides are sticking out into space. And that's really critical for replication because DNA polymerase uses those hydrogen bond donors and acceptors from the nucleotides to know what nucleotide to add to make the opposite strand. You have to have a single strand so that DNA polymerase can tell what the complementary nucleotide of the nucleotide it's reading, so to speak, is, so that it knows how to synthesize the second strand. So what is this going to look like a little bit later in time? What happens to the replication bubble as replication starts and proceeds? It's going to get bigger. And those enzymes, the helicases, are the enzymes that do the unwinding, the breaking of the hydrogen bonds between the two strands. So a little while later, the replication bubble will be a little bit bigger, with the rest of the chromosome still hydrogen bonded to itself. And this is about the point, for example, of the exercise that I developed for the last class, or we talked about last class, is at some point here, you'll see some RNA primers, depending on the polarity. I'm not drawing on polarity on here for a reason, just to make it a little bit clearer. So red RNA primers, you'll see some synthesis of new DNA starting at the 3' ends of those primers. And then replication proceeds. The replication bubble gets bigger and bigger as those second strands of DNA are synthesized. So that's the replication bubble. It grows in size as replication proceeds, and pretty soon, I guess I might as well take it all the way to the end. And I should caution, what's an oversimplification I'm making here about eukaryotic chromosomal replication? How many replication bubbles should there be on this chromosome? Usually more than one. Is that enough information on origin of replication for review, or is there anything else about replication, origin bubbles?
New DNA nucleotides. Does it start from the 3' end of the original strand, or does it start from the 3' end of the primers?
So DNA synthesis adds nucleotides to the 3' end of the primer.
Okay.
We're going to take a look. That's the next thing, as you can see in the slides, that's the next thing we're going to talk about. So yeah, we'll get there. 3' end of the primer is where DNA polymerase starts connecting new nucleotides. So right before the chromosome, if there was only one replication bubble on the chromosome, right before synthesis is complete, this is what, in a simplified format, that chromosome would look like. Those initial primers would have primed or enabled the synthesis of the leading strands. And then on the lagging strands, you'd see a bunch of short primers that each have initiated synthesis of the lagging strand. And then hopefully from this drawing, it might be easy to tell that what happens is once the helicases get to the ends of the chromosomes, the ends of the chromosomes are unzipped, and then you've got a full two copies of double helix. So on top here, we have, this is all double helix. The new strand is red and blue, RNA and new DNA. The old strand is black, but that's one double helix. We've almost made a complete copy of this whole chromosome at this point. And once the DNA polymerases reach, here are polymerases, once the polymerases finally move to the ends of the chromosomes, then both strands have been completely replicated. You've gone from one double helix at the top to two at the bottom. It's like the soundtrack to my life. Okay. So here's the Socrative activity. I'm just going to take a quick look at results. It's complicated because you can select multiple choices. So most people said A and D. We had a lot of B and C as well. So this is probably hard to do. I'm glad I'm not in your seats, because when I was designing this, I thought it's going to be really hard to stare at this on the quiz and not be able to actually label things and keep everything straight. So here we can go through it together. How do you figure this out? You start with one piece of polarity information, you can work out everything else. So on the bottom strand, the 3' ends on the left, which means the 5' ends on the right. And then the top strand's antiparallel. And the primers are antiparallel with the strands they're base paired with. So that's the 3', 5' ends, and the opposite on the bottom strand. So where is the first nucleotide going to be added? It's going to be stuck onto one of the ends of the primers. It's either A and D or it's B and C.
D.
It's A and D. That's where RNA polymerase is. It grabs onto the 3' end of the primer, and then it adds the nucleotide that is complementary to whatever is on the template strand. So if there was a T there on the template, let's use blue. If that was a T and that was a G, then the new nucleotides that come in are going to be C there and A. That would be the first DNA nucleotide that's added to the end of the bottom primer. This whole primer that's diagrammed, no letters, is RNA. Everything between C and D, in other words. That primer is all RNA, ribonucleic acid. That first A is DNA, like we discussed at the end of class last time. What we wind up with after replication is these little primers that are all still RNA, and then a lot of DNA that's been newly synthesized. And the last steps of DNA replication are you have to get rid of the RNA and replace it with DNA, which is the role of an enzyme we haven't talked about. That's okay. And then you have to have DNA ligase that comes in and bonds all of the backbones together. There's a key. In case you wanted to see the whole explanation, the DNA replication bubble labeling, it's in the slides. So you've got that for your reference if you want it. Okay, little activity. These are three, again, this is going to be, I think, maybe for now anyway, the end of origin of replication bubble analysis. But there are three different anonymous student submissions from this project that I would like you to please help me evaluate. So turn to a neighbor or a small group, pick A, B, or C. These are in bigger formats on the next three slides. So if you're looking at the slides, you can scroll to the big picture. You don't have to try to read these little icons. Figure out if there are any discrepancies between what we know about DNA replication and what is drawn in these three diagrams. Take 3 minutes or so, discuss with each other, convince yourselves that you are right, and then we'll get back together and we'll go through and evaluate each of these three and see what's right and what needs to be corrected. Okay? So it should get pretty noisy in here. Go. Are they right or are they wrong?
Yeah, they should be nucleotides. Yeah, there we go.
Who reviewed A? And was it okay? Were there some issues? What can we do? Was there anything wrong with this at all? Okay, so you could put in where the helix, that wasn't necessary, but where are the helicase in this case? Right, so the helicase will be sitting on the replication forks, moving in opposite directions. So they'll be unwinding the DNA that direction. So that's good to know for DNA replication. What else? Okay, so in this case, which direction, let's focus here on the top primer. This is the 3' end of the primer, and that's where replication is going to start. And that makes that the leading strand, as was correctly labeled here. But you don't want, so there would be no primer put there on the leading strand, because what would happen is DNA polymerase would start adding nucleotides on the leading strand. It would run into the end of a primer. So by definition, that's not what happens on the leading strand. The leading strand gets synthesized from the origin of replication continuously with no primers in the way. So here, the primers would need to go back here somewhere on the lagging strand. Anything else? Yeah, so the primer is, yes, that's a good point too. Hadn't caught that one. So the primer sequence here says TA, TA, and of course, we hadn't talked about that in class when I gave this assignment, but absolutely correct. The RNA would use U's instead of T's. B groups, what did you come up with?
B primers are not labeled with U's instead of T's.
Okay, so same concern here. The RNA primer sequences should have U's instead of T's.
It didn't identify the leading and the lagging strands.
Okay, leading and lagging strands aren't labeled. But we've talked about that a bit some, so now, on the last slide in particular, now we know where those go. Other comments? I have to try to remember why I put this on here. So this is showing the helicase. That's nice, pointing in both directions.
It put nucleotides on the outside of the bubble.
Okay, so that doesn't matter to me, but absolutely right. How you label where the nucleotides are is a critical concern to me. Whether it be on the inside or the outside, in other words. What about group C? Who worked on these ones? What did you find? We've got the RNA primers. We've got U's this time instead of T's. Is there anything at all? Going once? Going twice? Yeah, this is a good, well-labeled example. It doesn't have everything, but what it does have is accurate. And if you have any questions about the version of this assignment that you submitted and you want individual feedback, please let me know. This is sort of generic feedback. We covered now a lot of the things that I saw in most of the submissions. If there were problems, it was one of those things: primer placement, the direction of synthesis, or more minor point, U's instead of T's. All right. So getting into the extension material reading. This is going to be all about how scientists actually figure out where origins of replication are actually on real chromosomes, not just cartoons that I draw of chromosomes. So hopefully you took a look at this and read through the little bits of the paper that I asked you to look through. So specific paragraphs. And so I just want to highlight what I think is important about the abstract, for example. So this abstract, as all abstracts for all scientific papers, is just a brief summary of what is in the big 8, 9, 10-page long behemoth of a paper. So what's relevant here? First, some origins are highly active where others are dormant and rarely used. In other words, our chromosomes have lots of places where they start replicating. We've talked about the idea that there are multiple origins of replication. But the odd thing is that some origins of replication, some spots where our chromosomes start replicating, are always used every time every cell starts to divide and DNA has to be replicated. Other origins of replications are sometimes used, but not always used, which makes it harder for scientists to figure out where exactly the origins of replication are. Some of them are high efficiency or highly active. They're always used in every cell every time synthesis starts. But there are some regions of chromosomes that maybe it's sometimes an origin of replication and sometimes replication doesn't also start at that spot on the chromosome. One of the questions was, what species are these authors studying? So what species are they studying?
Budding yeast species?
These are yeast, Saccharomyces, and they're looking at 4 different species. So this is the main goal of this paper is you look at 4 closely related species, yeast just because they're easy to work with in the lab, and what they want to know relative to this idea that some origins are used frequently and some origins are used not so frequently is, if you look at 4 different species, are the frequently used origins of replication always frequently used in these 4 different species, or does it vary from species to species, where some replication bubbles start in one spot in 2 of the 4 species, but in another spot in the other 2? And presumably, this helps us figure out how to determine where the replication origins are. It's easy if you know that in all 4 species, it's always at this place. Presumably, but we'll see by the end of the discussion, presumably, the origin of replication is entirely defined by a DNA sequence. I think that's what people probably suspected, was there's going to be a particular unique DNA sequence that is the origin of replication sequence. So maybe keep that in mind as we work through the next couple of slides. Why does this matter? Well, here it is. Failure to activate, that is, failure to activate enough replication origins on the chromosome. We talked about the idea that the reason we have multiple origins is because our chromosomes are so long. We need lots of origins of replication to synthesize our chromosomes in time. When our cells are dividing, you can't take all day, literally, to replicate the DNA. When you're growing, your cells need to divide. But the cells that we have in our bodies, you'll learn about this more in 103, cell biology. Our cells have checkpoints, they're called. They're proteins in our cells that monitor the progression of DNA replication. And if DNA replication hasn't finished, then those proteins prevent the cell from dividing. So our cells are actively monitoring whether or not DNA replication is completed. But if some chromosomes, sometimes, occasionally, that checkpoint mechanism doesn't work properly, and the cell starts to divide before chromosomes have finished replicating. And that's bad. That's one of the ways we get aneuploidy, chromosomes that are not segregated properly into cells. So you get a cell that's missing a chunk of a chromosome because that part of the chromosome hadn't replicated when the cell divided. So there are human health issues related to understanding how origins of replication work and why sometimes they might not be used. So that's sort of the background motivation of this research study, was to try to figure out, if anything, does an organism know or decide which of its origins of replication to use? So this bit gets a little bit at the fact that there's a particular motif. We've talked about motifs. We've talked about consensus sequences. So this is where in the introduction, the authors are telling us this idea that I mentioned already, that there's a particular DNA sequence, maybe, that defines the origin of replication. And it says a match to this motif, whatever DNA sequence it is necessary but not sufficient for origin function. It means you have to have the sequence to be an origin, That's necessary. If you have this sequence, you can be an origin. You have the opportunity to be an origin of replication, but that's not enough. There's other things. It's not sufficient. There are other proteins that you also have to be an origin of replication. And then we mentioned this briefly a class or two ago, the idea that some origins of replication start replicating early in cell division, and other origins of replication create replication bubbles later during synthesis. And that's what you should take out of this paragraph. Some origins activate early in S phase, synthesis phase, and others later. So not only do some origins get used all of the time, and some origins only get used rarely, but they also differ depending on when during replication of the cell that origin fires. That is, the origin starts replicating DNA. Which is also kind of weird. You'd think all of the origins of replication should all start early in cell division so that there's enough time to synthesize all the DNA. But that's not what happens. So how do they tell where the origins of replication are? This is a pretty cool approach, so I've just explained it briefly. Imagine that you have a chromosome, and it's got multiple origins on it. So there's the chromosome. And what the researchers do, this is related to one of the questions. Let me see, I can go read the question that I asked. So, question 3, if a chromosome does not have an origin of replication, what happens to it?
It doesn't get replicated.
Fair enough. Half a point for the obvious answer. So let me go a little bit deeper then. Let's say you're a single-celled organism and you have a linear chromosome. We'll make it blue this time. So the cell's going to divide. What happens if that chromosome doesn't replicate? What happens to the chromosome in the 2-cell stage? Where is it? Yes, exactly right. Thank you. Well, it's going to go somewhere. Does it get cut in half and eat half of the chromosome goes in? Right, no. So it's going to go in one or the other of the 2 daughter cells. Why is this a problem? Hopefully it's a problem.
Because the cell can't survive without it?
Because cells have to have DNA, because the DNA encodes the genes and the proteins that make the cell function. Right, if you have a cell, actually, we do have cells that don't have DNA. We'll talk about that later. Does anyone know? Red blood cells. So we do have cells that don't have DNA, but it turns out red blood cells are they're a terminal cell. That is, they've been created to do their job. They don't need DNA to do their job. So they're a rare example of a cell that does not have a genome. But most cells need their chromosomes to survive. In other words, that would not be a viable cell. So what they do to figure out where the origins of replication are, let's diagram those as little green bubbles. If there are origins of replication across a linear chromosome, what the scientists do is they break the chromosome physically into chunks, and then they move one of those into a plasmid. That would be like a circular chromosome that bacteria have. So each of these different plasmids has a different piece of DNA from a eukaryotic, a linear chromosome. So in this case, each of those 2 pieces of the black DNA, the source DNA, has an origin of replication on it. But there are also times where breaking the chromosome into little bits will cause production of those little stretches of the host chromosome that does not have a replication origin. So then all they do is they put each of those artificial chromosomes, the plasmids, the circular chromosomes that have a little tiny bit of the eukaryotic genome cloned into them, cut and pasted into them, is they'll put each of those into one of these cells, like a bacterium or something. And if that chunk doesn't have an origin replication on it, no origin, when this bacterium divides into 2, that chromosome won't have been replicated. It will only go into one of the 2. And the scientists can detect that. Right? They look at all multiple cells and they figure out which cells have this chromosome we put in. If there is an origin of replication, then what happens? If that little bit of eukaryotic DNA is an origin of replication, when the cell divides, then that circular chromosome will also be replicated because you have to have an origin of replication to replicate the chromosome. So it's a functional assay. You test the ability of a small piece of chromosome to drive the replication of a chromosome. And that's how these scientists determined where on the yeast chromosomes the origins of replication were, this sort of approach. So in this case, they would find that bits of the genome that do have an origin of replication will pass on to all of the daughter cells, and then you can detect that. So that's what they did. One type of experiment, determining mutation. Skip that one. Okay, so those were all of the questions that I'd asked you to look to answer based on this paper. So we already talked about the ARS. What does ARS stand for? Did anybody pay attention? That was the easiest. That was the easiest one. ARS ACS. What does ARS stand for?
Replication site?
The R is definitely for replication. We're a third of the way there. Okay, thank you. Autonomously replicating sequence. That is completely related to the previous slide. They're looking for pieces of chromosome that are able to replicate other pieces of DNA. Autonomously means by themselves. So it's a piece of DNA that by itself can cause chromosomes to be replicated. So basically, ARS equals origin of replication. And what's the ACS? The ACS is if you take a bunch of ARSs, like ARS1, ARS2, and ARS3, you collect the sequences of different pieces of chromosomes, all of which can act as origins of replication, and you do what we've done before and make a sequence alignment and look for a consensus. So that's what the C in ACS stands for. ACS is the ARS consensus sequence, ACS. Take all of the ARS sequences, you line them up, and let's say one's ATAC, one's ATTC, one's ATAG, you get the consensus sequence ATAC. For example, that's not the actual ARS sequence. So that's what the authors did. They broke the yeast genome into a bunch of bits, they tested which parts of which chromosome can act to drive replication, and then they found out the sequences of those pieces of DNA. They found whether or not, this is, again, the question they were asking, do they all have a common DNA sequence? Do all of the origins of replication in yeast have some sort of DNA signature that we picked where origins of replication are? So at last, we get to the one figure that I asked you to look at. There's a lot going on. This is common for scientific papers if you haven't read many in your life, which you may not have. So there are 5 different panels. Right? The 5 different, what we call panels, 5 different groupings, 5 different types of data in this panel A, panel B, panel C, panel D, panel E. They're all different. If you want to know what they are, the best thing to do is to look at the figure legend. The little bit of smaller font size text next to or underneath the figure that explains what A means, what B means, what C means, and so forth. And I think I asked you to focus mainly on panel B. So what's the, I don't know, what is this, like Christmas ornaments or something? What does this represent? This is a cartoon, it's not data, it's a representation of data. Horizontal line.
The origins of replication.
What's the origins of replication? The line?
No, the bubbles.
Okay, the circles represent origins of replication. So they're saying, so what's the line? Okay, so it's a representation of a chromosome, something about base pairs. So we're looking at a chromosome, a drawing of a chromosome from left to right, from 0 base pairs to, in this case, they're looking only at 270,000 base pairs, 270 kb, thousands of base pairs. So you're looking at a small piece of a chromosome, and each of the bubbles represents an origin of replication. So this part of this chromosome has 1, 2, 3, 4, 5, 6, 7, 8, 9, I don't know, something like 13 or so origins. What's related to the size of the bubble? What does the size of the circle mean?
How long it's been replicating?
How,
Long it's been replicating, or how old the origin of replication is?
that is a good interpretation. Unfortunately, that's not true for this, but close. So this, yeah, this might look like, this is not, what this is not, which I think is what's misleading, is like my drawing of a chromosome where we're looking at the size of replication bubbles. And that would be an origin that just fired on the right, and this would be an old origin there. In this case, the circle size represents how frequently that origin is used to start replicating DNA. Big bubble means high frequency. Big circle means they're almost always used. The little circles are the ones that are very rarely used. So if you looked at 1,000 yeast cells and their origins of replication, maybe the biggest circles are used 100% of the time, which would mean that one's used 66% of the time, and maybe those ones are used 33% of the time, the smaller ones, something like that. Is there anything striking to you? You don't need to know anything about it. Is there anything interesting about the pattern of the sizes of the circles or where they're located on the chromosome?
They're more bigger in the middle.
They tend to be larger in the middle. Okay, that's true. What about big versus little? If you're an origin of replication, if you're one of the big circles, one of the origins of replication that almost always get used, then would you want them to be spread apart or clustered together? Right, because if each of them is always going to start replication, if you want to replicate your chromosome quickly, you'd want them all evenly spread out on the chromosome so that all the parts of the chromosome get replicated at about the same time. You wouldn't necessarily have them close together, which is a little bit odd about the middle of the chromosome. If you are always used all the time, then if you're an infrequently used origin, if you rarely get used to start replication, where are you more likely to be found, do you think? Near another origin or far from another origin? If you're an origin that rarely gets used,
You'd be near another origin.
then you probably aren't very necessary. So something has to replicate the chromosome, and so you're more likely to be near a big origin or a frequently used origin. That is, all of the little dots tend to be close to other origins. And that's probably why they're infrequently used, because there's an origin right next door that always gets used. So if that origin here is always initiating replicate, well, sometimes, say 50% of the time maybe based on circle size, then yeah, nearby origins are probably less likely to also be used all the time. So origins tend to cluster in the middles of the chromosomes. They are spread out, but more likely clustered near, and the small, infrequently used origins tend not to be used very frequently. They tend to be near other origins. Last, for this figure, I think I also suggested looking at panel E. So what is panel E? What are they trying to show us? How do we interpret it? What are the three different lines? They're sequences from
The three different species used.
the three different species used. And what's the point? What do you think we're supposed to take away from the authors showing us these three sequences from those three species? What's in bold? What's shaded? Yeah.
Are they trying to show us like a consensus sequence?
Okay, so this is the ACS. This is the ARS consensus sequence from each of the three different species. Do we expect them to be the same? So remember, ARS consensus sequence, that's the average sequence for an origin of replication in each of those three different species. So we've got the Cerevisiae average consensus sequence on the top, and then another species and another species. Should they look the same? Probably. They're closely related species. They're origins of replication from these three different species. Do they look like they're the same sequences in the three species?
The top two look fairly similar, but the last one, the only thing that is similar is that small area with all the T's. That's
Yeah.
it.
Yep. So that's why they bolded those letters, because they're trying to point that particular motif out. That this ATTT, ATA, TTTT sequence in bold in Cerevisiae looks pretty similar to the origin of replication in Saccharomyces paradoxus, the middle row. So that's where the authors suggest, yeah, there is something about a particular DNA sequence that's almost always found at every origin of replication in that species. So you could predict, if you knew the ACS, by just looking at genome sequence, chromosome sequences, you could predict where the origins are going to be by looking for that sequence. But what they saw that was really weird is that when they looked at a more distantly related species, same genus, still Saccharomyces, but Saccharomyces bayanus, their origin of replication sequence, the ACS, doesn't look at all like the top two. It's totally different. It's still A's and T's, but it's mostly T's. It's not the same sequence. So that then makes it kind of hard to predict. You couldn't use, in other words, the Saccharomyces cerevisiae origin of replication sequence to predict origins in a different species. They're different. The sequences that define origins of replication are different.
Well, looking at E and looking at the Bianus,
Yeah,
is that
sure.
what you call it?
Yeah.
The Bianus species, and then also looking back up at B, and they're using both Paradoxus and Bionisis.
Yeah.
You can see that the Bionisis doesn't successfully use those same sites
So-
as often as Paradoxus.
Absolutely.
Which makes a lot of sense.
It's really, and so this is an evolutionary genetics paper. This is not pure genetics. They were interested in understanding whether or not the use of different origins would differ between species. So that's what the check marks and X's indicate. That this autonomously replicating sequence, the tiny dot there in between the two big origins, is only used in Saccharomyces cerevisiae. It's not used at all as an origin in these other two species. Which is really weird, because you'd think something as basic and essential as replicating your damn chromosomes would be conserved among species. That's a really important thing to have to do. So why would there be so much difference in how this process works from species to species? That's an evolutionary biology phrased question. But there's genetics underneath it, because there's DNA sequence that predicts where the origins are, but the origins change from species to species. There's some similarity, but there are some differences. And any questions? Concerns, gripes, criticisms, comments?
Does the origin of replication always, is it always the same area as a consensus?
So restate that question, please.
So does it replicate always at the consensus sequence?
So in other words, everywhere on the chromosome you would see that sequence, is that an origin replication? Is that what you're asking?
Sure, yeah.
Okay. That's a great question that I don't know the answer to. I will look it up and find out. Okay? I will report back, and if I don't, call me out on it. And it's in the recording, so now we've got proof.
the consensus sequence in figure E,
Yep.
it doesn't always have to be a palindrome?
So this is not, yes, so this does not have to be a palindrome. This origin of, in fact, that leads me to the last point, and then we'll talk about what to do for next class with the flyover. What's similar about the ACSs for all of these species? This bit that I've circled in red and then the line underneath it. In that region, what's similar in all three? Does it look like random DNA sequence, or is there some difference between those three bits and random DNA sequence in this region?
There are only A's and T's.
There are only A's and T's. Why is that? And is that relevant, or is that just chance? So, that brings us to the last question that I had posed when I asked you to do this reading. Question 5. In figure 1E, what is common to the ACS, and why would it make sense that this type of sequence would be an ACS? Why would those nucleotides be particularly useful to be an origin of replication? They're all A's and T's. What do we know about A's and T's?
They only have 2 hydrogen bonds.
A's and T's have 2 hydrogen bonds that hold them together, not 3. So if you were going to choose, if the cell had a brain and you were going to choose where on the chromosome you are going to unwind the double helix and start replication, what kind of sequence would you choose? One that's held together less strongly, more weakly, which is where there are lots of A's and T's and no G's and C's. So most origins of replication, regardless of what species you look at, are going to be highly AT rich. So I will do this briefly at the start of next class, just one more sequence alignment. There's one other feature of sequence alignments I'm going to add in this example that you should know about. And then at that point, we'll have been done with all of this. The one thing that I didn't talk about, I think, we've touched on all of these vocabulary items except for one, gyrase. So I'm going to show you gyrase right now, but this is what to do for next class. So finish that Canvas discussion board, read the next topic, and then I'm going to do a quick demo if I can have a volunteer.